Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology topics. This is cardiovascular health and risks. The first part is topic 1C, part 1, risk correlation and cause. A risk is the probability that an event could occur. Sometimes it could be underestimated or overestimated based on how familiar one person is with the activity. So if they've had relatives who have been smoking but have never had cancer, these people could underestimate the risk. Also, it depends on how much one enjoys the activity. Again, for smoking, if somebody enjoys it, or if somebody enjoys eating fatty food, they're not going to think of the risk being high in comparison to someone else or something else. Also, whether or not one approves the activity, of course, if they enjoy it, they're going to be able to underestimate the risk. In assessing risks of developing certain diseases, epidemiological studies can be carried out. Also, we need to know that some diseases are multifactorial, like cardiovascular disease, of course, caused by many factors. A correlation is a link between two sets of data, and it can be positive or negative. Also, a correlation can show how data changes. Further studies can be required to see if there is a causal link between the two data sets. In topic 1C2, we're going to look at investigating the causes of CVD. When designing scientific studies to see the progression of disease, it's better that these studies are bigger because those provide more reliable results. And of course, some studies are carried out for longer periods of time. These are called longitudinal studies. Most of the time, they study the same types of people for longer periods of time. So the results are going to be more reliable. Of course, in all scientific studies, one factor is going to be varied while others will remain controlled. However, when we carry out studies on humans, there are so many variables that are so hard to control. So sometimes results could be a little different due to the uncontrollable variables. In metadata analysis, massive literature studies are analyzed. Sometimes they're small or large data in the subject area to give more reliable evidence on that specific topic or one study that is being carried out. Next part is how do we evaluate scientific studies. Of course, we have to be very careful. We have to find out validity of the results, meaning, do the results answer the question that has been asked? The results have to be precise, meaning there is less deviation from the mean between the results of one specific study. Reliability of the results, can the results be reproduced by other scientists? Also, we have to find out who funded the research. This is to ensure that there is no bias. Also, where the results were published. If results are published in a journal where the peer reviewers are friends to the person who carried out the research, substandard results could be published due to the friendship between the two parties. Next, we'll look at 9 lifestyle risk factors of CVD. Of course, these include genes or DNA, they include age, gender, and so on. Some people are genetically predisposed to develop CVD. For example, some people's arteries easily get damaged. They have a tendency to have hypertension, as well as problems with balancing cholesterol in their bodies. Of course, the ratio of 3:1 cannot be balanced properly, or they do not break down cholesterol easily, and therefore that is going to be due to their genes, so it's going to lead to problems that could result in CVD. When we get to age, as people get older, blood vessels lose elasticity, and therefore they begin to narrow, and this can increase the chances of them getting CVD. When we go to gender, of course, statistically, men under 50 and women over 50 are likely to suffer from CVD. For women below 50, due to estrogen, they have a lower risk of developing CVD. However, after menopause, the risk of CVD increases. Sometimes twin studies are very useful in determining if a genetic or lifestyle factor has led to CVD, of course, because twins or identical twins do have similar DNA, so we can study if lifestyle factors are the cause of the CVD or genetics. Going to part 3, these are risk factors and cardiovascular disease. Of course, we're going to the lifestyle factors. These are many. They include smoking, inactivity, they include high blood pressure, obesity, and many more. So for smoking, of course, the chemicals within the smoke cause damages to the arterial lining, and this can lead to buildup of plaque. Smoking also causes arteries to narrow, and this raises the blood pressure, increasing the risk of formation of atherosclerosis, and that could lead to CVD. There is another lifestyle factor, inactivity, lack of exercise. So if you have regular exercise, this can help you lower the blood pressure, and it can prevent obesity as well as diabetes, because you're going to be using up the excess energy and it's not going to be stored as fat. So this is going to lower the blood cholesterol levels and balance lipoproteins as well as reduce stress. However, if somebody lacks exercise, of course, there is going to be buildup of unused carbohydrates as fat, and some cholesterol is not going to be metabolized, and that could lead to buildup of atheromas within the blood vessels. Next is high blood pressure. When somebody has hypertension, it can increase the risk of damages to the blood vessels, leading to plaque formation. The next factor is diet, which could of course lead to obesity. A poor diet can lead to much cholesterol building up in the body. It can lead to diabetes. And if this is coupled with inactivity, the risk of developing CVD can increase. Evidence from current studies suggests that eating a balanced diet with variety of fats and plenty of fruits and vegetables can help to prevent atherosclerosis. In part 4, we're going to be looking at how diet affects cardiovascular health. Here we're going to be considering BMI as well as waist to hip ratio as measures of the risk of CVD. BMI is also called the body mass index. Scientific evidence suggests that what somebody eats has a way it affects their health. And in our bodies, if we have excess food, this can be converted into fat and stored. And if not metabolized, some of this fat as well as cholesterol could build up in blood vessels. So BMI can be measured to estimate if somebody has a higher risk of developing CVD or not. BMI is equal to weight in kilograms divided by the height in meters squared. However, when we use BMI, it cannot tell the difference between fat and muscle. So those are the inefficiencies of using this method. And sometimes it underestimates body fat in older people who have lost some muscle. So due to these inefficiencies, BMI is not the best predictor of CVD, but if we couple it with other factors, it's going to be useful in determining the risk of CVD. The next predictor of the risk of CVD is using the waist to hip ratio. Of course, this method has advantages as well as disadvantages. To do this, we get the weight in centimeters divided by the hip size in centimeters. Of course, to measure the hip size, you have to measure the widest area of the hips, and then this is measured slightly above the navel. So you divide and get the waist to hip ratio. Now, the inefficiencies of this method is some people could measure areas where the waist is thinner instead of the actual areas they have to measure, and this is going to underestimate the risk of CVD using the waist to hip ratio. Going to diet and CVD, like we've already said, whatever somebody consumes can increase the risk of CVD. And there is a relationship between fat in the diet as well as cholesterol in the blood. Remember, if you eat too much fatty food, there is a chance that a lot of cholesterol is going to build up in your blood vessels, and this could increase the risk of developing CVDs. However, when we look at this, we have to talk about the LDLs as well as HDLs. Low-density lipoproteins, or LDLs, these are made from saturated fats. These should be fats, as well as proteins. These bind on cell membranes before being taken into the cells. However, if the cell is saturated with cholesterol, more LDLs will remain in the blood, meaning they cannot be taken up by the cells, and this could build up in the blood vessels. When we go to high-density lipoproteins, of course, these are mainly from unsaturated fats, proteins, as well as cholesterol. These carry cholesterol around the body to the liver where it's going to be broken down, decreasing the amount of cholesterol in the body. The HDLs can even remove cholesterol from fatty plaques to reduce the risk of CVD. You need to remember that a healthy LDL to HDL ratio should be 3:1. Next, we go to dietary antioxidants and CVD. There is some evidence that suggests that antioxidants are good at decreasing the risk of developing CVD. This is because they neutralize or remove free radicals. If free radicals are not removed, they cause damage to the endothelial cells, and this leads to inflammation, and in response, plaque formation is going to occur, and this will lead to CVD. There is some evidence that suggests that food rich in vitamin C have greater antioxidant properties. However, there is no conclusive data. Expect questions about vitamin C case studies, and they can also ask you about the core practical of investigating the presence of vitamin C in a food sample using DCPAP. This is usually going to be in Unit 3, but it can also be in Unit 1. Remember, DCPAP is a blue solution that is decolorized if vitamin C is present, but it's going to turn pink if acidic solutions like lemon juice are the ones tested for vitamin C. In topic 1C6, we're looking at using evidence. Of course, a lot of scientific evidence suggests that non-lifestyle and lifestyle factors can lead to CVD, so it's better that people prepare, or it's better that people change their lifestyles because it's more expensive to treat a disease than preventing it. There is evidence that millions of smokers die from CVD as well as cancers every year. So if people change their lifestyles like stop smoking, this could decrease the risk of developing CVD among many people. Now, because the body mass index as well as the waist to hip ratio may not be that efficient, there are other methods that are used, like how much oxygen somebody takes in during exercise. This could be a better indicator of somebody's heart condition. In looking at why people don't change their lifestyle, of course, some people enjoy it, some people are depressed, and so on. And also, some people can distinguish between a perceived risk and an actual risk. They assume that the risk applies to somebody else. I can use an example of somebody whose grandparents have been smokers, but they did not die of cancer. If they begin to smoke, they will also assume that they will never die from cancer due to smoking. However, when somebody gets cancer, then they are scared and they will take it seriously. Another reason as to why people don't change is they underestimate the risk of CVD associated with smoking, obesity, high salt intake, as well as lack of exercise. Also, some people smoke as a way of maintaining a lower body mass because when you smoke, it speeds up your metabolism and reduces your appetite. So some people lose weight when they smoke, and they prefer to smoke in order to lose weight. Most people who are smokers are addicted to the nicotine, so sometimes they use electronic cigarettes to get nicotine into their bodies but to prevent the chemicals or toxins that come with cigarette smoke. In part 7, we'll look at the benefits and risks of treatment. Here we're going to be looking at medications that can be taken. For high blood pressure, we can take antihypertensives. These include diuretics. These eliminate excess fluid to decrease the blood volume, as well as salt. The next one is beta blockers. This blocks the heart response to hormones such as adrenaline, which work by speeding up the heart rate. The next one, we go to sympathetic nerve inhibitors. These are going to inhibit the action of the sympathetic nerve. The sympathetic nerve is the one that speeds up impulses to stimulate arterioconstriction. So the sympathetic nerve inhibitors are going to block this effect to prevent arterioconstriction. The others are ACE inhibitors. These block the production of a type of angiotensin. It's called angiotensin 2. This is a hormone that stimulates blood vessel constriction, leading to a raised blood pressure. Also, angiotensin 2 stimulates the sympathetic nerve, causing the blood pressure to increase, and inhibits the parasympathetic or vagus nerve system, and in turn increases the blood pressure. So if this hormone is not produced in high amounts, this means blood pressure could go down. Next is controlling blood cholesterol. This can be done using statins, plant stenols or sterols. For statins, these reduce or lower blood cholesterol by blocking the enzyme in the liver responsible for making cholesterol, or LDL cholesterol. They're very effective at blocking LDLs, and they improve the balance between LDL to HDL, and this reduces inflammation in the arteries. However, they could ask you about the side effects. Remember, in some people, statins trigger a form of muscle inflammation that could be fatal. So this is one of the disadvantages of taking statins. Going to plant stenols and sterols. Of course, these reduce the amount of cholesterol absorbed from your gut into your blood. Lastly, we go to anticoagulants and platelet inhibitory drugs. For anticoagulants, the example we have is warfarin. This interferes with the production of prothrombin, and if there is less prothrombin, then it means thrombin cannot be produced, and therefore fibrinogen cannot be converted to fibrin, so there is going to be less blood clots. A dosage of this anticoagulant is going to be monitored closely to prevent excessive bleeding. The other ones are platelet inhibitory drugs. These include aspirin. They make platelets less sticky, reducing the clotting ability of the blood. So for people who have higher risks of formation of blood clots that could lead to other complications like CVD and so on by sticking in the blood vessels, that could be minimized. Since we looked at aspirin as the example, we have to look at the risks of aspirin. These include irritation of the stomach lining as well as bleeding within the stomach. Again, this bleeding is because there is reduction in clotting ability of the blood. When they use this kind of medication, it's difficult to achieve the correct balance between preventing blood from clotting too easily while allowing the clotting to occur when necessary. So it means sometimes people could bleed excessively in different areas of their bodies, and it could also lead to other problems or complications that were not foreseen. So they have to be closely monitored by a healthy expert. So this brings us to the end of this topic. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.